Okay, so 29. We're on the RNA, the creation of RNA and the different RNA polymerases. So the eukaryotic promoters are going to differ depending on the type of RNA polymerase. The RNA polymerase 1 promoters sequences will include the ribosome initiator element, that is the R. Now this is I. Oftentimes people mess this up, and it's very common for you to hear this. So be a little careful when people start saying these quickly, okay? So RI initiator, IN, right? R. This is going to be at the transcriptional site. The upstream promoter element, which is UPE. The RNA polymerase 2 promoter sequences are going to include a set of consensus sequences. These are going to define the start site and recruit the polymerase 3. The RNA polymerase 3 is going to act as the promoter sequences. It's going to locate within the transcribed sequence downstream of your start site. This will include the type 1 and type 2 promoters. And so this is actually what this looks like in terms of figure. And can you guys even see that? Yeah, that one's okay. All right, so right here we have the RNA polymerase 1 promoter. And so we have here the upstream promoter element. Everybody understands what upstream means? If you look at this, it's like a flow of a stream, right? It's ahead of it. Okay. And then this one here, that is actually this right here, which again is the ribosomal initiator element right there, okay? And so it connects around this point. What's then going to happen is this is going to then start transcribing on down. And that's what we're seeing right there. See the little arrow? There we go. Now, this is going to be the RNA polymerase 2 promoter. And right here we have an enhancer and then a TATA box. This TATA box right here is going to be part of the promotion. It's going to be your upstream promoter element that's going to start that promoter right here. Okay? And so this again is going to be the initiator. Okay. And this is the other way this can go. You can have the no TATA box here. You can then have the promoter has a DPE region right here. And this is going to cause promotion to start right here. And this is where actually your initiation occurs. Okay? Now, both of these cases right here, these enhancers will come back and they'll enhance that transcription activity. Okay? Remember that this is not actually a road or something that's fixed. This is a polymer, right? Like a rope. It can wrap back on itself and touch, okay? I used to have a really good video that showed this process. I cannot find it anymore, unfortunately. I have no fucking clue what happened to it. Probably a sacrifice on the 500 gigabytes of material I got rid of. Yeah. The RNA polymerase 3 promoter, this is that right here. We have two types within this. Type 1 is going to be the single-stranded RNA. Remember, the single-strand RNA does not have to be double-stranded, right? RNA very often can be single-stranded. And so this is going to have the A and C block, and those need to be near each other. Type 2, this is going to be for the tRNA. This right here is going to have the A block, and then we can have quite a bit of separation, and then we can have the B block, okay? And that's what that means whenever you see this double, that it has a fair amount of separation, okay? It doesn't usually have to be something super specific, and it can be a pretty wide range of separation, right? We just didn't want to actually say how much it is. When you see something like this, these actually have to be quite close to each other, okay? At least when you're talking about like DNA, right? Okay. So your RNA polymerase 2 promoters is going to be generally located on your 5' side of your start site for the transcription. They're called the cis-acting elements, this because they're on the same molecule of the DNA as the transcribed genes. And so this is the TATA box. This is usually found between positions -30 and -100. What does -30 and -100 mean? Yeah, it's the number of bases that you'd be ahead, because it's negative, right? So think about it like this as starting line, and then the first starting position in a race, you have the second one that's a little bit further back, third position a little bit further back, and so on, okay? So this is going to be 30 to 100 positions back from that starting position, upstream. The mutation of a single base is going to markedly impair the promoter activity. And so this is right here is a TATA box. The important part that gives us its name is, guess what? TA. Okay? Is this on the DNA or RNA, by the way? Has to be DNA, right? Yeah. Couldn't be RNA because RNA would have had? Yeah. Exactly. Okay, and so that's your 5' going to 3', TA, and then the rest of this just needs to usually be like adenosine rich. So this right here, by the way, these are the percentages that you will see these numbers, okay? So for example, the TATA has an 82% chance of this T being here, okay? That does mean 18% of the time it isn't there, right? Okay. And this 97%, 93%, 85%. What about this one right here? It's 50-50, right? You very oftentimes do not have an A there, okay? And that's because we've already completed the TATA, right? The initiator element, INR, this is actually from the previous figure, INR right there. Okay, this is going to be found at the transcriptional start site. It's going to be between positions -3 to +5, and that's why we had two different ones. We had to represent that, right? Sometimes ahead, sometimes behind. Okay, sometimes upstream, sometimes downstream. This is going to define the start site, the downstream core promoter element. This was the DPE. I'd kind of side dodged describing what it was before. This is going to be found downstream of the start site. It's going to be positioned usually around +28 to +32. Again, that means downstream when we say positive, right? Because that's further into the reading. Okay, so commonly found in conjunction with the INR and transcripts, this lacks the TATA box. We do not have a TATA box. So the sequences of the CAP box and the GC box are going to be highly conserved as well. Additional regulatory sequences are located between -40 and -150. Everybody's comfortable with the negative and positives now? Okay. What would 0 be? Yeah, typically this right here, right? Now remember, we don't actually necessarily always start exactly on that position, the INR, okay? Where it actually ends up starting would be 0. And then every one after that would be, +1. One behind that would be -1. So the promoters that contain a CAAT box and some contain a GC box, constitutive genes, the genes that are going to be continuously expressed, tend to have GC boxes in their promoters. And this is a GC box, by the way. Typically, it's just repeating GC units. The CAAT box and the GC box can be effective when present on the template antisense strand. This right here being the CAAT box typically has a pretty good prevalence of G's, C's, and T's, by the way, in it. But that's the CAAT box. So the transcription factors, the proteins that bind to these cis-acting elements to regulate gene expression, transcription factors for your RNA polymerase II are going to be collectively known as TF, transcription factor, TF2. Which one does it act on? 2. I know, every now and then you're reminded that biochemists actually do pay attention to their terminology and are careful about it. Yeah. Go figure, huh? Individually, they're known as TF2A, TF2B. Guess what the next one would be? Yeah, you got it. Which is beautiful. I actually really prefer that over when they use the alpha, beta, gamma. Yeah. I think that actually makes a lot more sense and it's easier to remember for people because a lot of people don't do the Greek alphabet enough to really get it down. The TATA box promoters, the TATA box binding protein, which is TBP, T for TATA, of course, B binding protein. This is going to be a component of your TF2D. This will bind to your TATA box. Okay, so this is your TATA box binds to the concave surface of your TBP. This is going to induce that large conformational change into your bound DNA. Okay, let me ask you something. If you want to like straighten out sheets and stuff, or you want to work on like rope or something, what do you normally do? Do you just let it wave around everywhere? No. Like you're folding a sheet, what do you do? Yeah. You grab it, bunch it, so you can hold on to it, and stretch it out, don't you? And so this is kind of very similar actually, isn't it? Basically, it's pinching a part of the DNA. See that? Yeah. And changing that DNA conformation so it's no longer wound up like it used to be. And it's no longer flopping everywhere and having issues, right? So we've actually done a conformational change of the DNA by essentially pinching it. Can you see that? Yeah, come on, you gotta appreciate that. So the general transcription factors are going to be essential in your initiation of your transcription by the RNA polymerase II. Your TBP saddle is going to provide the docking sites for the binding of an additional transcription factors. Your pre-initiation complex, known as the PIC, this is going to be your complex that will contain these elements, which are TF2A, B, F, RNA polymerase II, TF2E, and H. I know, hell of a complex, right? All right, now this right, can you guys even see that figure? Well, okay, let me. God, I forget sometimes exactly the projectors I'm using. I'll be kind of honest there. Better? Okay, so this right here again is your TATA. This is your TBP. What does that look like again? So it looks like a saddle is what they call it. And if you look, I don't know how much you guys even are exposed to horses. How much are you exposed to horses? Or anything you'd put a saddle on? How about motorcycles? Yeah? Okay. Motorcycles also have saddles, right? Okay? Okay. So can you recognize the saddle? Yeah. We're going on that. Here's our bases. Like that. DNA is wound like that. We're saddling across it. Okay. And it goes across almost, not quite perpendicular, sort of 45-ish degree-ish. A little bit more than that. Okay. And so we're saddling there with our TBP on that TATA. This is then going to have the complex assembled around it. And so this D right here, which one's that? This is going to be our PIC complex, in case you're wondering. Okay, so we're going to assemble our PIC complex around here. And again, this is going to be the different parts assembling around it. And you can actually see the different parts in here on their labels. These right here, the smaller ones, will be these right here. The TF2, and we have the A right there, the B, F. This is why that order, by the way, was kind of weird, when I listed them, in case you're wondering why I listed it like that. Then we have the E and the H right there. This big yellow thing, can anybody tell me what that is? You got it. Yep. That's the RNA polymerase right there. That's what's going to be attaching and starting off at that INR site within, a few bases of that. Okay. And so why does this, so we assemble the complex on there, and then it's going to have the DNA is going to open, your CTD is then going to be phosphorylated, and then you're going to actually lose the majority of this complex. Everybody see that? Okay. This part's saddled on the DNA, so that's going to be left behind. Okay. This part here is going to be the polymerase that's going to go along and do your copying, right? Why does it have this string of stuff coming off it? Phosphates on it. What does it have to take in to be able to do this process? NTPs, right? Yeah. And then this is your string coming out, right? Yeah. And notice it started with it coming in, nothing coming out. And so we've done this for a bit now. Okay, note what stays attached to it, by the way. TF2, you got it. Yeah, the B, E, and H all come off. Okay, which is kind of interesting because the F actually starts off coming on after the B, right? So this part gets left behind. This part, all of these fall off. Only that remains attached to the polymerase. Yeah, it's actually part of that complex that will do that transcription. Yeah. So it's your full machinery is not actually a complex in and of itself. Much like the DNA polymerase, we talk about DNA polymerase doing the copying and doing that process, but it's the complex that actually does it, right? Okay. So the TF2D, this is going to be the one that recognizes a core promoter elements. It's going to be central to the assembly process. And this is that TF2D, of course. And that's going to be where it actually recognizes it, and we actually end up attaching the TBP to that. The TF2A, this may aid in your binding of your TF2D to the DNA. And so this is going to come in. And, I don't know if you guys would even know this. Sometimes, do you guys know of a rope climbing lock? No? Yeah. Okay, you're at least familiar with the idea of something could slide along, right? And it could recognize and attach. And to make sure it attaches properly, you need usually something to clip it down, right? Yeah. And so this is very much what this is doing. It's going along here. It matches well, so it's energetically favorable. And then we have another item that comes in and clips. And that's going to basically secure that site and make sure it matches really well, right? And that will then identify all that machinery together, identifies this site, and promotes binding of the rest of the complex to this site. So then you can start at your proper transcription site. Does that make sense? Just go around in a circle. Okay, so at this point we're bound to our TATA site right before our transcription site. The TF2F, this aids in the recruitment of your polymerase 2. I missed the B. The DNA binding protein that recognizes the B recognition elements near the TATA box is TF2B. So TF2B binds first. This is the one that's going to recognize that we have that binding complex ready. Okay? Once that's recruited and bound, then we're going to start assembling the rest of this. And that's the TF2F. The TF2F is going to make the RNA polymerase 2 here bind more readily to that site. By the way, the TF2F is actually the one that would end up basically where the starting site would be. The initiation site, the 0, that's the one that's going to end up basically there. Okay. This is why, by the way, you have to be like 30-ish down, is because this machinery takes up space, right? So if you were too close, you'd actually go over your site. You couldn't actually assemble on it. Your TF2E, this is going to be what brings the TF2H to the complex. TF2H, this has helicase activity. This is going to be what unwinds your DNA. And it has kinase activity that's going to phosphorylate the CTD of the polymerase 2. This will facilitate the transition to the elongation. Okay, so once we have these things attached, at this point, right here, we actually have our transcription complex attached, don't we? Why the fuck do we need E and H? I mean, look at this. It just gets rid of it, right? Okay? Okay, how do you know when you're ready and how do you get started? Could you transcribe this when it's all wound up? What do you need to do to start transcription? You have to unwind it. And so that's what this is doing. This is recruiting your unwinding agent. This is going to unwind it. This is also going to help you recognize that you're actually ready to go and elongate that, that polymerase 2. So it's actually in its reactive, fully functional form. Okay. The enhancer, this is going to be the cis-acting element. This is going to increase the activity of many of the promoters in the higher eukaryotes. We have no promoter activity but can stimulate promoter even over distances of thousands of nucleotides. These get pretty weird, actually, and they're kind of cool because it's a lot like it wraps back in on itself, and then it hits the point every now and then. And then if the complex is there and the promoter hits it, it's like a little gun going off, right? Because these are actually very fast if you think about them as base per second. And then it starts going, whoosh. Again, these can be upstream, they can be downstream, they can be in the middle of the transcribed gene. As long as they can wrap back around and be detected, they're good. It doesn't really matter where they are, right? Because is there anything different of the bottom of a rope coming back around and hitting it versus the top of the rope coming back around and hitting it? No, there's no real difference, right? Doesn't matter. They're effective when present on the coding or non-coding strand. Again, it doesn't matter because they wrap back around and make contact. They're effective only in certain cells, though, because you do have to have the machinery to do it. Virtually all transcription products are going to be further processed in eukaryotes. Some prokaryotic transcriptions are going to be modified as well. Many bacterial mRNA molecules are going to be translated while being transcribed. Because remember, they don't have nuclei, right? And they don't need further modification. So literally, as that little piece of RNA is coming off, that mRNA, it just feeds directly into ribosome. And then you start translating it while you're still in the middle of transcribing. It just needs to be long enough to get over to the ribosome area. Bacterial tRNA and rRNA molecules are going to be modified. Transcripts can be cleaved at specific sites along the sequence. Nucleotides can be added to the terminae of the RNA chains. Bases and ribose units of the RNAs can also be modified. So this is actually the cleavage of the primary RNA transcript in the E. coli. It's going to produce a 5S, a 16S, and a 23S rRNA molecules, and a tRNA molecule with tRNAs right there. So right here we have the 16S, a 16 segment of this. This right here gets excluded, gone, tRNA, and then we have our next RNA part. This is a 23S, and this one's a 5S right there. This is uridylation residues. They're going to be modified after the transcription to form the ribothiamidylate, pseudo-uridylate. So again, this is uridylate right there. And we are modifying this. This one has a CH3. This one right here is the nitrogen here instead of just, it moves one space over for the nitrogen. Yeah, basically rotating nitrogen. The RNA polymerase 1 produces 3 ribosomal RNAs. The RNA polymerase 1 synthesizes a large precursor RNA. This is the 45S unit. That's a subsequently processed to yield the 18S, 28S, and 5.8S rRNAs. Did you ever think you'd see a 5.8? No, yeah, so we actually clip off a little bit of extra. The 18S are RNA. What are the rRNAs again? Yeah, they're ribosomal. Is ribosome just going to be RNA? No, it has protein in it as well, right? And it's going to assemble the ribosomal complex. Okay, the tRNAs, they're just tags, right? They're small little fragments of RNA. And then you have the mRNA, and those are going to be the one that we actually are going to have the code for our protein on, the messengers. Okay, so we have the 18S RNA. It's going to be the RNA component for the small ribosomal unit, subunit. And so that's going to go on to the 40S small ribosomal subunit. Again, that's with protein and everything. The 28S and 5.8S rRNAs are going to be your 2 RNA subcomponents of the large ribosomal subunit, which is 60S. The pre-rRNA, again, is going to be this 45S unit. Here's the 18, 5.8, and 2 28S units. This is your nucleotide modifications, by the way. These ones get methylated, and so those are your methyl groups going on. And we have the pseudouridine as well. The pseudouridine typically denoted by that Poseidon psi symbol. Okay? And this is what you got right there. And those will be these modified units right there. Okay, so the processing of the 45S precursor RNA, step 1 is going to be the ribose and base components of your nucleotides. They're going to be extensively modified. They're going to be directed by the RNA protein complexes, as we call these small nuclear ribonucleoproteins, or SNORNPs for multiple. The step 2 is going to be the pre-rRNA. This is going to be assembled with the ribosomal proteins. This is going to form a large ribonuclear protein. Step 3, the rRNA is going to cleavage. This will release your mature rRNAs. These are going to be assembled with ribosomal proteins. These will then form your ribosomes, which will then form your ribosome complexes. Again, here's the rDNA. Right here we have the pre-rRNAs coming off of it, right there. And then right here we actually have the processing, and so this is the SSU processome. These are the ones that we'll actually see. You can see one attached here. See that? Yeah. And that's actually what's processing it. Okay. So for both the eukaryotic and prokaryotic tRNAs, which ones are the tRNAs? Transfer, yeah. They're the tags, right? They're going to tag the amino acids. So they're in a readable form for the RNA code. Okay? So again, eukaryotic and prokaryotic tRNAs, the 5' leader is going to be cleaved by RNase P. the 3' trailer is actually removed. The CCA is added by the CCA adding enzyme. In eukaryotes, the tRNAs are going to be heavily modified on base and ribose moieties. Many eukaryotic pre-tRNAs are actually spliced by an endonuclease and a ligase to remove an intron. So right here we see the leader, the 5' end leader, and right here this is the early transcript of this intron right here, with the intron right there. And then we have right here the OH, which is the 3' end. And see it wraps back around. Anybody know what this is being held together by? Base pair matching. There's hydrogen bonds. Okay. And so what we get here is the leader actually gets processed off. We have a P attached there. That's that 5' end. Okay. The intron is actually going to be removed here. This is going to leave us with the anticodon here, which would match the codon on the mRNA, right? Okay. And this right here was then going to get processed to the amino acid attachment site. Notice that this is a change from the previous ending. See that? Yeah. Okay. And then what exactly does that do? That gets attached to the amino acids. They drift in. If this anticodon happens to match well with the codon, then the ribosome can attach that amino acid, right? So it'll be positioned correctly. If the anticodon is not matched properly, what happens? Yeah, it'll end up getting rejected, right? Yeah. If it matches just barely well enough, what would happen? We could change out the amino acid. Yeah. Okay, so the product of the mRNA polymerase 2 and the pre-mRNA transcript, this is actually going to acquire a 5' cap and a 3' poly A tail. The pre-mRNA is the immediate product of the RNA polymerase 2. The pre-mRNA molecules are going to be spliced to remove the introns. They're going to undergo modifications to your 5' and 3' ends. Keep in mind that I'm an mRNA now, no longer tRNA. So the 5' cap, remember that cap, head, right? Lead. Okay. Tail is the ending, right? Okay, so this is going to be a distinct terminus at the 5'. This is going to contain unusual 5' to 5' triphosphate linkage. It's going to protect the 5' ends from the phosphatases and nucleases that will contribute to the mRNA stability, so you don't end up cutting it away and processing it and then end up with just a part of an mRNA. Okay? This is going to enhance the mRNA translation by the eukaryotic protein synthesizing systems, i.e., anybody ever seen like a feed tape? Did you guys ever see like the old projectors? Okay. What was on the tip of the old projectors? Like the beginning, leading strand. Nobody knows. Yeah, it was junk stuff, but it wasn't just junk stuff on the film. The important part was there were little grooves. And so that was literally the cap, okay? It was stuff that didn't really matter if you saw it, and then it was stuff that would attach well to the machinery, okay? And that's what you're getting here. The poly A tail, on the other hand, this is going to be a string of 250 adenine nucleotides at the 3' end. This is going to be added by a poly A polymerase after the cleavage of the pre-mRNA by the endonuclease. This may enhance translation efficiency and the mRNA stability. It does it through a few different methods. Again, that's that OH. That tends to be one of the more reactive parts of the molecule. And so, you know, if something attacks the poly A tail, not big deal, right? You got extra A's. What else is important about that? What is 3 A's for the codon? Does that say keep making more protein and adding more amino acids? Yeah. Yeah, it's the stop. The 3 A's would be the codon. And so the poly A tail is repeatedly saying stop, 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 stop. Yeah. And again, it doesn't have to match exactly, right? Because you're doing lots of A's all after another. It doesn't matter what frame you get. Okay? So you could screw up and add an extra amino acid, but the next one's going to say stop, isn't it? Okay. And by the way, that's the exact same thing as the film example I gave you. If you were to stay at the end of the film long enough, you would just see repeated frames of junk. Okay? Sometimes other films, by the way. You know that? Yeah. Yeah. They would sometimes splice extra parts of other films. So deleted scenes that they literally delete scenes by cutting them. And if you were, out of money and you needed to make that tail on your, they would just take parts of whatever happened to be on the floor and tape them in. And so when they made copies of those reels, they could actually end up with stuff in those. Yeah. And it happened more than you'd think, too. So, anyways, filmography, interesting stuff. The 5' ends of the eukaryotic mRNAs, again, are going to be capped. This is that same cap we've been talking about that's going to feed in to our ribosome. The 5' triphosphate end is going to be immediately modified. So we have a phosphoryl group that's going to be released by hydrolysis. The diphosphate 5' end is going to attack an alpha phosphorus atom of GTP. This will form our 5' cap. The M7 nitrogen of the terminal guanine is going to be methylated. This will form the cap 0, and is going to be, and is going to be adjacent riboses, may be methylated to form cap 1 or cap 2. Okay, and so right here actually we have this, see that nice little GTP here, okay? And then this is going to be methylated cap 0, 1, and 2. This is methylated in cap 1 and 2, and this is methylated in cap 2. And that's the methyl group on that second oxygen. Why do we have that second oxygen there, by the way? Because this is the RNA, not the DNA, right? So this has the OH normally, and that's why we're methylating those. This is going to help make this more stable. What's one of the reasons that we go DNA instead of RNA? It's more stable. Why is it more stable? OH is next to each other, oxygen's next to each other, tend to be more reactive, right? So if you methylate those, you protect those. Okay, so the primary transcripts in your eukaryotes are going to be polyadenylated. So here's your template DNA, and right here we have our nice little transcription, and we have the nascent RNA coming off here. And then what's going to happen is this right here is going to be our cleavage signal, and that's the AAU, AAA. So next what is going to happen is we'll have our endonuclease attach, and this is going to start cleaving things that are not going to actually be expressed. And so what's then going to happen after that, we've cleaved out the stuff we're not going to express, we add a poly A tail using a bunch of ATPs. And so we add AAA a bunch of times. Okay? This typically adds about 250 adenylated residues. So this is actually quite a lengthy tail. Remember though that this is a full gene can be thousands of residues long, right? So it's not necessarily as long as you might think. Most genes in the eukaryotes are going to consist of your exons. Again, those are going to be your expressed or coding regions, and introns, which are going to be the non-coding regions. The RNA splicing, this is going to be a process through which the introns are excised and the exons are going to be linked. This will form your final mRNA. Your introns can vary in length from 50 to 10,000 nucleotides. They can be exceptionally long. You can have fragments of a functional protein and enzyme, which are separated by 10,000 length fragments. Yeah. And so you just end up cutting all that away. Sometimes these can be one gene's introns could be exons into something else. Okay? So don't think that you're necessarily only making and expressing one gene when you do this. Okay? You could actually end up assembling an entirely different mRNA from the stuff you cut out of one. Right? The common structural motifs of the introns are going to have common structural motifs, which are begins with a GU and ends with an AG. The consensus sequences of the 5' slices and vertebrates are going to be AGG, UAA, GU. The first GU is then going to indicate the start of your intron. The consensus sequence at the 3' end of an intron is going to be a stretch of 10 pyrimidines. This is called the polypyrimidine tract. It'll then be followed by an NCAG, with the AG indicating the end of the intron. The branch site is going to be located between 20 and 50 nucleotides upstream of your 3' splice site. This is it all together. And so right here we have the intron. What is the intron again? Is it going to be included in our gene expression? No. It's the incised, right? Removed. Okay? Okay, so this right here is the 5' splice site, 3' splice site. This is where the actual cut happens. So the expressed portion has what? That AG, doesn't it? That's actually part of the exon. Okay? So your AG, GU, this right here is the end. This part actually gets included into the intron. Okay? And then we have a bunch of stuff here. This has branching sites here. That's our intron. This right here is the 3' slice site. One of the important things is we actually have the pyrimidines here. That's that repeated pyrimidine-rich region. That's going to indicate, hey, pay attention to me. I'm getting to the end of an intron. Okay? It actually physically makes a different structure that comes back and binds on itself when you have these expressions like this. Okay? It's not actually like a, I know I use the film example a lot, but it's not like a film. Okay? Because remember, it has stickiness to each other. It tends to wrap on itself and bind, right? And so it's more like, has anybody ever seen the shamans that stored information with the knots? Or the storytellers? Have you guys ever, who here's Catholic? Or knows Catholic stuff at all? Anybody? Okay, so Catholics have the rosary, right? And the different beads actually have meanings. And then you can have a story of God, from the Bible usually, expressed within the rosary, right? And so it physically has a change in the structure that tells you things about it. And this is very similar to that, right? Okay. Was that a better example? Okay. Maybe rosary will work. Okay. So anyways, this is the downstream. This actually splices at that GG. And so the GG, the G part right here, actually is in the downstream exon. Okay? All right, so the spliceosomes is going to be large complexes of small RNAs and proteins where the splicing is going to occur. Not just the ribosome is a complex of RNA and protein. Spliceosomes are as well. Most of the stuff that directly works with the RNA-DNA process, the transcription process, is actually RNA-protein complexes. Probably because RNA was the original thing that all the viruses and different organisms that predate was using. So the splicing begins with the cleavage of your phosphodiester linkage. This is going to be between the exon 1 and the 5' end of the intron. The attacking group in the trans-esterification reaction is going to be the 2' OH of the adenylated residue in your branching site. And so this is what we actually get here, is we had a diphosphate linkage. Remember, these are quite reactive, right? Because we have all these oxygens attached to that phosphorus right here. So here we have a lone pair. This can then attack that phosphorus, causing this to become a leaving group, right? And that's what we get. And that's referred to as trans-esterification. Okay? You may recognize that from our trans-esterification reactions we kind of touched on in organic chemistry. Okay? Now, the 3' OH of the exon 1 is going to attack the phosphodiester linkage between the intron and exon 2. This will join the 2 exons with another trans-esterification reaction. So right here we have our precursor. Here we have our exon 1, exon 2. This is our 5' splicing site. This is our 3' splicing site. Notice we've expressed the phosphodiester linkage. See that? We've put the P in little circles so we can see it. Okay, now you may have been wondering why the hell the branch site, right? Remember this? Branch site. Okay. And specifically an A, right? Okay. Now, that's actually because of this right here. So the branch sites, that A, that's the OH that's actually going to attack. Okay? And it's not the normal 5', 3' OH that's going to do it. It's a branching reaction. So it's going to be the 2' OH that does it. And that's the one that's going to do the trans-esterification off of the 5' end. And so you go ahead and get the trans-esterification. It transfers this phosphate to attaching there. This is now freed from the 5' end on that G there. And this right here is then going to swing around and it's going to attack up here on that phosphate. Okay? And so that's how we actually collect these exon 1 and 2. Right. Everybody sees that okay? Fantastic. Okay, so what that's going to leave you is the exon 1 bound 3' end here onto the 5' over here. And this is going to give you your splice product, of course, which is going to go on and get expressed. This right here is the lariat. Everybody's seen a lariat before? Tie the rope back on itself and then, woo. Yeah. Okay. 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 And it looks like a lariat, right? See, it's tied back onto itself. Yeah. All right. So this is that lariat form of your intron right here. And again, we have the full intron, and it has now been removed. Okay. Does anybody have any questions about that? Okay. So the trans-esterification reaction will generate the branched and forms that lariat. Again, that can also be called the loop intermediate. Oh, I'm out of time. Okay, that is the week. You all have a wonderful weekend. And next week is the final week of lectures. It's also the last exam will be on Friday. If you want to try to take the exam early or anything, that's fine. Go for it. I'm happy to have you come in during office hours sometime. I'll sit you down and you can do it, okay? Otherwise, don't forget, finals are coming. Check that final schedule to make sure that there's no issues, because they don't have a final time for our class schedule, which is not supposed to exist, from 11 to 12 on Tuesdays and Thursdays, right? It's so if you go to the class schedule here, I'm actually going to go like this. Damn it. Okay. And go down to finals. This is my current final schedule. There's few enough of you that I really don't care if I do this on exact schedules. Okay? So if you guys want to change any of these dates, I am absolutely fine. You just contact each other, talk about it. Mm-hmm. We can. You just work together to figure out a time, okay? The easy way to do it, by the way, is if you guys use Google Calendar, you can actually have a possible dates, and it'll like match the calendars and show you where they're open. The Tuesday one, I'll actually be giving another final at that time. So technically, because of the scheduling and the time slots that are supposed to be available for those, like we actually have 2 classes that are in the normal schedule for me. Yeah. So my schedule's weird. But what I'll do is I'll have you guys just come in here with the other class and do it. You all take care.